Hello and welcome to lecture number 18. Today we're talking about DNA replication. Okay, let's recap what we discussed in the last lecture. So the last lecture was the one about DNA inheritance. We talked about inheritance, and then we talked about linkage and X chromosomal or sex chromosomal linkage as well. And so what we discussed in that second lecture of the Mendelian genetics or genetics portion we talked about biological sex and gamete size and how that is defined. We said that sperm producers are often referred to as male and egg producers as female. And we discussed that gender refers to a self-defined identity and that gender and sex are not the same thing. And then we talked about sex chromosomes and sex linkage. We said that in animals with XX and XY sex determination, just like it is in humans, recessive genes on the X chromosome show up in the XY individuals. We talked about genes that can be linked to each other. I said that linkage and sex linkage is not the same thing, and to make sure to keep that apart. And then linkage analysis was discussed with respect to how to map genes relative to their distance to each other onto a so-called genetic map. And at the end, we talked about the inheritance of plastids and mitochondria. All right, so today we will talk about how DNA is synthesized. We've been talking about how it's inherited, how entire pieces, chromosomes, are inherited, and what the significance of that is, which of course is that the genes are located on the DNA, on the chromosomes. But we haven't discussed how DNA is replicated. Now, think back to the beginning of the semester. We did discuss how RNA is made from DNA strands, where DNA strands code as the template for the RNA molecules. And so that is what is relatively similar to the discussion that we'll have today about DNA, as DNA is being synthesized as a new strand from an older DNA strand template. So in general, what we will be looking at is what's called a replication fork. So this is a piece of DNA. So you have your two strands of DNA, and they are, of course, held together by the hydrogen bonds between A's and T's or G's and C's. And then as it turns out, and we'll talk a little bit about how that was found, DNA, when it's being synthesized, is first unwound. And so you get this fork where the replication occurs, and that is what's called a replication fork. And then each parental strand, so each of the dark blue strands, now serves as the template for the synthesis of the new daughter strand, which is the light blue strand. You see that that's not here yet, right? It just gets added into here or here as the DNA is replicated. So how do we know that DNA replication occurs like this, where each strand provides the template for the new strand and is being made at the same time? So in other words, every old strand gets a new strand as the two daughter strands are being made, as opposed to the other possibility where the DNA is unzipped, read, and then closed back up again, and the new copy is completely independent of it. So that was the question that Meselston and Stahl were trying to analyze. And you may remember, or we may not have discussed it in much detail, that when the structure of DNA was first proposed by Watson and Crick, in their short paper that they wrote, in which they announced their model, they also said at the end, this model suggests a way of replicating DNA, because when you unzip a piece of DNA with two strands, then you have one strand that immediately can synthesize, that can be used as a template to synthesize the other strand. But that hadn't been shown. And so what was done in this landmark experiment by Messelson and Stahl is that they used a technique where they added what's called heavy nucleotides or heavy isotopes. So they used isotopes, and you might remember from the beginning of the semester that isotopes have additional neutrons in their nucleus. And then we have the radioactive isotopes that are unstable and they can fall apart and that can lead to energy emission and so forth. But the point here is that these people were using the same elements, but slightly different versions of those. And so those are the isotopes. So you can distinguish different isotopes from each other because of the weight of the difference between them in their weight. And the reason for why they're different in weight is because they have additional neutrons. And so those neutrons weigh something, and that adds to the difference between one isotope versus the other isotope. So what did they do? So at the beginning of the experiments, both strands are labeled with heavy nitrogen. So this is 15N, that's heavier than the normal nitrogen. And the way that they do that is just that they grow bacteria in solution. And so they have a bunch of bacteria. They have their regular normal nitrogen in their DNA. And then they take the bacteria, they strain them or they centrifuge them, get rid of the liquid in which the bacteria grew, and then put them in new liquid in which they can go in nutrient broth. And in that nutrient broth, they now add the heavy nitrogen. So that means that as bacteria continue to grow in the new medium, only the new DNA will incorporate the heavy nitrogen if there's no light hydrogen available. So all the old stuff will be labeled with the light nitrogen, all the new stuff will be labeled with the heavy nitrogen. And so after one round of replication, in the absence of heavy nitrogen, the parental strand still contains the heavy nitrogen. So they then took them back out and put them in light nitrogen. And then they looked after one round of replication to see whether or not we now had two strands of heavy and two strands of light, or whether they had one strand of heavy and one strand of light. So what the book says here is that after one round of replication, in the absence of heavy nitrogen, the parental strand still contains the heavy nitrogen, but the daughter strand contains light nitrogen. So you see the dark and the light blue molecules here, signifying that. The daughter DNA molecules form a band with an intermediate density. So what that means is that once they did this experiment, they then took aliquots out and they centrifuged them. So they put them in these centrifuge tubes and they put it in a medium that is thick, like a thick substance, thick liquid though. And so in that thick liquid, the DNA would settle depending on its weight as the tube was spun in the centrifuge. So round and round it goes, and the heaviest stuff moves down to the bottom of the tube faster than the lighter stuff. And so you can see here, here's the heaviest stuff. The intermediate stuff is sort of in between. So they tell you here about the density of the DNA. 1.7 is the highest that moves the lowest in the tube. 1.715 is a little higher in the tube. It's a little bit lower in weight. And then the lighter one with two strands of light DNA labeled with a light nitrogen, the 14 nitrogen, is only 1.708. And so that will determine whether or not whether it settles in that tube higher or lower. And so then after two rounds of replication, half of the duplex DNA molecules have one strand of the heavy and one of the light. And the daughter DNA molecules therefore form two bands, one intermediate in density and the other one light. So we start with the heavy stuff. We have the intermediate in the first round of replication where we only have the dark and the light. And then if we go to the next round of replication, now one light will give rise to another light one, and the other dark will insert another light one, right? So you will always have this one heavy molecule, but all the future replication molecules will become light. And so you can see that in the next round doing the centrifugation. Okay, so what's important to know about this experiment? It was conducted with different isotopes. The heavy one sinks further down. And the two possibilities that they originally wanted to distinguish was the one that where the original template unzips, the new strands are made, and the original template then just rezips. And so there's an old strand, double old strand, and a double new strand. And that would have been called conservative replication. But that's not what happened, right? You see that this occurred. It didn't happen like this particular hypothesis. The other hypothesis is what was shown to be true. And so in that hypothesis, they suggested that the original template unzips, each strand codes for a new strand, and each daughter molecule contains one new and one old strand. And that's what you see here, right? One new, one old strand. That is called semi-conservative replication. So if the question is, well, does DNA get replicated conservatively or semi-conservatively? The answer is semi-conservatively, because one strand is the old strand and the other strand is the new strand. All right, so now let's take a look and see how these strands are made. So I want to remind you of the time when we discussed RNA synthesis. At that point, I tried to emphasize that the RNA strand always grows from the 5' to the 3' direction. So remember what that means, how the sugar molecule is counted, the carbons in the sugar molecules are counted, 1, 2, 3, 4, 5, and that the 5' end versus the 3' end on the bottom is where the addition of the next nucleotide occurs. And so that's the same as you have in the case of DNA replication. The new strand always grows from the 5' end to the 3' end. It starts at the 5' end, grows to the 3' end. And because of the anti-parallel nature of DNA, where the 5' end of the other strand runs into the other direction, right? Remember that you have 5' strand here, 5 to 3 here, and 5 to 3 in this direction on the other strand. That has to be done on the new growing strand as well. So you have the DNA strand coding, serving as the template here, and the new strand as it's being made, being synthesized from the 5' to the 3' direction. So incoming nucleotides are accepted if they correctly pair. Of course, we have the GC and the TA, and the 3' hydroxyl of the growing strand attacks the high energy phosphate bond of the molecule as it's being added to the growing strand. And then that initiates the synthesis reaction. Okay, so this shouldn't be too different from what we've seen before with the difference that now we're looking at DNA synthesis, not at RNA synthesis. So, but there's a problem, because we have two strands that we have to deal with now rather than just one strand that we're making, right? We're not just making one single-stranded RNA molecule, we're making a double-stranded DNA molecule. And so that's now problematic, because the DNA is not only read from one of the two strands, as it is for RNA synthesis, it's read from both strands, and both have to be copied. So what happens is that the DNA is synthesized in two directions at the same time. So if you think this is a piece of a DNA, if you imagine this is it continues over here and it continues over on this side, but we have a piece of DNA that is currently growing, and you have the 5' end over here and the 3' end over here on the top strand, and on the bottom strand the 5' end is here, the 3' end is here. So if the DNA polymerase can only extend the DNA from the 5' to the 3' direction of the new strand, of the growing strand, then that means that the top strand can only grow from here to here, right? It can only grow in this direction. But on the bottom strand, it can't grow in this direction, because that would mean adding nucleotides from the growing strand's 3' to the 5' end. And the DNA synthesis, the DNA polymerase cannot do that. So in order to make it happen anyway, what happens during DNA synthesis is that this strand starts at the point where the DNA is being unzipped. But the other part, the other strand, is synthesized in the other direction, starting a little bit further away. So the process starts here and it moves into this direction. So you have your two strands that are growing from this end inwards and from that end outwards. Now, this molecule down here, as you continue to unwind this piece of double-stranded DNA, this could just continue to grow, right? Just continues to grow and grow and grow and grow. But this one will continue to grow into the other direction. And then what happens to the part of the top strand as it's trying to be synthesized? So that would mean that as this piece is being elongated, and this piece is being elongated, but sort of just continuously moving inwards into this replication fork, that hole has to be filled. And that hole is filled by simply adding a new start over here and starting in this direction. So you have one continuous growing and one so-called discontinuously growing strand. It starts here, continues to grow, but then you start anew from over here and you continue to grow until you reach the point of the piece of new DNA that was put down already. Okay, so that is called discontinuous versus continuous growth. So you have the continuous growth on the bottom strand and the discontinuous growth on the top strand. And the terms for those strands are therefore called the leading strand. This bottom strand is the leading strand because it leads the synthesis. It just continues onward, while the other one always lags behind a little bit. It doesn't lag behind very much, but it just lags a little bit behind, enough to call it the lagging strand. Okay, so we have the directions, we have the leading and the lagging strand, we have the pieces that you have to make versus the continuous piece. But how do we start this whole process? And so that's where this primer comes into play. A primer is just a starting point. So this is the place where the reaction starts, and it needs to be primed with a piece of nucleotides. And it turns out to be not DNA nucleotides, but RNA nucleotides. It's probably a relic from old evolutionary times. But it's a piece of RNA, and it's red here. So that is a piece of RNA. It binds to this part because it fits the sequence, right? So it's complementary to this piece. It's complementary to that part. So that little primer is built, is added, and then that's where the DNA synthesis can bind. It needs something to bind. It cannot just bind DNA. It needs this primer to bind. And when it binds that, then it can sit down and it can read the nucleotides as they are being unzipped in this direction and as they are presented to the DNA polymerase here in that direction. So if there's a G over here, then it will put in a C. If there's a T over here, it will put in an A, and so forth. And the same, of course, in the other direction. So we have elongation in the two different directions. And we have the primer placement, and at the end of the time when this new piece is being added, and it comes in contact with this primer, then that primer is replaced, so it gets cut out. We'll talk a little bit more about it in just a sec. It gets replaced, and then this piece gets finished, and the two ends get sealed. So we'll come back to the details. So just as an overview, just for you to see that there's a leading and a lagging strand, that we have the problem with the two directions that we have to synthesize because the polymerase can only synthesize the new strand in the 5' to 3' direction. So remember that DNA molecules are antiparallel. Remember that the enzymes build DNA and RNA from the 5' to the 3' direction of the growing molecule, and that one strand here can grow continuously, but the other one has to be built in sections. Okay, so now let's take a look at this a little more step by step. We'll zoom in a little bit and see what's exactly happening, what enzymes are involved, and what the individual steps are. So I already told you that a RNA primer has to be put down, and that gets done by an RNA primase. Remember that if you see a molecule name that ends in ASE, that's almost always an enzyme. So anything that ends in A's is most likely an enzyme. So the primase is the enzyme that puts down the primer, the piece of nucleotides, that has to be complementary to the DNA here. Then you get the DNA polymerase, so again an enzyme ending in A's. Now this makes DNA, that's why it's called DNA polymerase, means making long pieces out of short pieces. So it continues to grow this piece. And then when the two sections collide with each other, then there's an enzyme, and your book doesn't mention it here. I'll write it down in a second. It's called an exonuclease. Exo means out, nuclease means to cut. So it cuts out that piece of DNA. And then that's what it says here. The primer is removed, and then a DNA polymerase molecule comes in, finishes up that little piece that was taken out, those 10 or 20 nucleotides. And then in the end, there's an enzyme that's called ligase that joins the two fragments together. So the ends are just basically sealed using that ligase. So good to remember would be that we start this process with an RNA primase, that's an enzyme. Then we're adding an RNA primer, that's a building block of RNA nucleotides. And then we have these two enzymes, the polymerase and the ligase. Okay, so that's the process with which you have the growing of these strands. But we need a lot more, right? We need to take the DNA. We have this big ball of stuff that's really compact, and we need to unwind it. And we, if you have something that's twisted and you just unzip it, then it gets more and more compact at the end, right? Think about two strands that are tied around each other, and you just pull them, they just get tighter and tighter, unless there's something at the end that either twists it in the other direction or somehow relieves the stress between these two intertwined strands that otherwise just get tighter and tighter wound. And so we have enzymes that do all that. Let's take a look at that. And I write down a few things here in a second. So if you look at the replication fork, again, this is our replication fork. We're getting DNA being made here. We have this big ball of DNA that needs to be unwound so that we can then unzip it. We have two enzymes that help with that process. One is called the topoisomerase, topoisomerase 2. Your book just says it relieves the stress of unwinding. So basically the way it does that is by making little nicks into the DNA and then sort of twisting them around. And not so important that you know that right now exactly how that works, but it essentially nicks and reseals the DNA at the end so that it doesn't get tighter and tighter and tighter wound. And then the second enzyme is called the helicase, and the helicase essentially melts the DNA apart, right? We remember, maybe you remember, that DNA is bound together with its hydrogen bonds because A's and T's and C's and G's bind, and in order to get that not to stick together, you would have to put in energy. And the energy is, you could just melt it apart with brute force by heating it up. And DNA would eventually melt apart. The two strands would melt because the energy in those hydrogen bonds is broken. Well, that doesn't work so well in cells. So cells have enzymes to overcome that activation energy, right? To make that less energetically costly, or it doesn't require the heat in order for this melting to occur. And that enzyme that does that is called helicase. So that unwinds the two parental strands. So now you have single strands here, single-stranded binding proteins. They stabilize this DNA, so basically hold it apart. Think about two pieces of tape that you have stuck together. Now you're pulling them apart, and the longer that piece gets, the more you have to be careful that if you release any kind of tension, that it doesn't sort of fold back on itself and gets all stuck together. So that's what the DNA, the single-stranded binding protein does. So it keeps these two sticky ends apart from each other, and then the DNA polymerase, that's a complex of multiple subunits, binds and acts at the site of the growing chain to increase the chain length one DNA subunit at a time, checking for errors. So it's making errors, but it's also checking those errors. We'll come back to the proofreading capability in a little bit at the end of the lecture. But you see how the DNA polymerase just continues to grow in the leading strand. It just chugs along and puts in one nucleotide at a time. And the same thing is true for the DNA polymerase over here. Same enzyme, by the way. I mean, not the same exact molecule, but the same kind of enzyme, and it moves into the other direction. So individual steps that you should remember are the DNA unwinding requires the topoisomerase, the unzipping requires the helicase, the RNA primase lays down the RNA primer. We have the DNA polymerase that extends the DNA. We have the DNA polymerase on the other end that extends the small bits. And what I haven't told you so far, but I'm telling you right now, is that those little bits also are called the Okazaki fragments. Okazaki, for the Japanese scientist who first found out about this. So in his honor, they're called Okazaki fragments. Then the RNA primers are removed by this enzyme called exonuclease. I said I was going to write it down. Here it is in writing. And then filled in by the polymerase. So this is DNA polymerase. It's a slightly different polymerase, but it's essentially polymerase that does the same thing as this polymerase. And then at the very end, these Okazaki fragments, every time that an RNA piece is put out and filled in, then has to be sealed, and that's where the enzyme ligase comes into play. So this would be good to know. So proofreading. So that's it's kind of interesting that this enzyme has in its ability not just one function, but multiple functions. So it binds, it recognizes the nucleotide on the other side. It sees, for example, an A and knows that it can now only fit in a T, and it does that as one function. But it then also sort of pauses, looks at what it's done, checks whether that actually fits well, and if it doesn't, it stops. And so that's the proofreading capability. So if you look here at this replication fork, there are two replication forks really at a long strand of DNA. If this is a chromosome, you can have the replication fork in multiple spots, and then that is called a replication bubble. So if we just zoom in on this and we look how the DNA polymerase chugs along here, and it puts in an incorrect nucleotide. That doesn't happen very frequently, but when it happens, the proofreading function removes and replaces that piece with a correct piece. Your book has a little animation for that. I might as well show you that.
In DNA replication, each daughter strand is elongated step by step by adding successive nucleotides to the 3' end of the growing strand. Each nucleotide added to the growing strand contains a base that undergoes proper base pairing with its partner in the parent strand. The base A pairs with T, and the base G pairs with C. Very rarely, a nucleotide with an incorrect base is added to the growing strand. This improper base pairing activates an alternative capability of DNA polymerase called an exonuclease activity, which cleaves and removes the incorrect nucleotide from the 3' end of the growing strand. After the incorrect nucleotide is removed, the polymerase activity of DNA polymerase is restored, and the correct nucleotide with a properly paired base is moved into place and attached. The process in which DNA polymerase removes an incorrect nucleotide and replaces it with the correct one is known as proofreading. After proofreading removes an incorrect nucleotide, the enzyme's polymerase activity returns and synthesis continues.
So you heard her say the term has exonuclease activity, right? So that is when, in addition to its polymerase activity, it also exonuclease activity uses exonuclease activity. Exo meaning cut out, away from, and nuclease means cutting DNA, so it cuts the DNA out. That's what it can do in its proofreading capability. So mutations in DNA polymerase that don't have the proofreading capability anymore because of a mutation, they will just make a bunch of mistakes, and that will lead to a lot of mutations. And so that can be a big problem if that capability is mutated. Okay, so let's take a look at these bubbles and the forks that I just mentioned on the previous slide. So in long molecules of DNA, as chromosomes are, it's just one molecule. So humans have 23 times 2, so 46. So they basically only have 46 molecules of DNA, but they're very, very long. And so if you were to replicate that chromosome just from one end to the other end, that would take very long. And so that's not how it works. You start the replication process at multiple sections, and those are called origins or origins of replications, and oftentimes simply abbreviated as ORIs. So the replication can begin at any origin of replication, which is a specific DNA sequence. So it doesn't start anywhere, and then wherever it starts happens to be an origin. No, there are certain DNA sequences that can be recognized by the machinery, the enzyme machinery, and only at those particular sequences, which are called origins of replications, can an origin for a new replication fork start. And so then each replication bubble has two replication forks that move in opposite directions. And so this one will move in that direction, and that one will move in that direction. And at some point, two of these bubbles will meet, and they just sort of pop together, and then we have the long strands, and they get fused together with the enzyme ligase. So that's how organisms with linear chromosomes do that. Small microbes mostly that have circular DNA, circular chromosomes, as well as circular non-chromosomal DNA. We haven't talked about that yet, but I'm just telling you that there are also little pieces of DNA sometimes that are non-chromosomal. They're not really part of the main chromosome in bacteria. Those are called plasmids. You will be working with plasmids later on in the lab. That's why I'm mentioning this here. They are circular. So they're circular DNA, oftentimes found in bacteria. The main chromosome is often circular in bacteria, and there are plasmids, which are small extra genomic pieces of DNA, also circular. How do you replicate a circular piece of DNA? Well, they also have origins of replications, but there's something easy about that, and that is that if it's a circle, all you need to do is start somewhere and then go around on both ends, and then at the end you pop together, and that's the end of it. So you only need one origin of replication, and then you get the two replication forks and produce the new double strand. So circular DNA molecules usually only have exactly one origin of replication. All right, so there's one more thing that we need to consider when we think about how DNA is replicated on a long molecule, on a long linear molecule, and that is the ends. So if you think about what I told you earlier, that a RNA primer has to be placed down, and it happens to be placed down before the DNA strand is unwound. So in the fork, just at the end of, or however you want to see that, where the fork starts to open up, that's where the primer gets put down. Well, that means that if you look at this replication fork here, that piece is still stuck together, right? There is nothing here yet where the RNA polymerase, where the RNA primase could put down the primer. And so therefore it doesn't. And if it can't do that, if the last primer that gets placed is only close to the end but not at the end, then you always end up with a little piece of DNA that's not getting replicated. That piece is this piece up here, right? It wasn't, it's only the top strand that we're looking at. See, this is only the top strand we're looking at. The bottom strand is now no longer pictured here. And that's because the bottom strand has no problem, because that leading strand can just zoom all the way out, and then when it runs out of DNA, it falls off. But the top strand, the lagging strand, cannot be replicated fully to the end that way. And so you end up with unreplicated pieces of template DNA on the lagging strand. And so in the next generation, this would be the end, and then in the next generation, this would be the end, and it would get shorter and shorter and shorter and shorter with every replication cycle and with every cell division cycle. So after a few rounds, you would be potentially missing the genes that are at the very end here of the chromosome. So I say that's the problem of finishing the lagging strand. And I write down here, just in summary, what that is, that the end of the lagging strand is not completely unzipped when the last RNA primer is laid down, and that leads to the fact that the last few bases of the lagging strand are not replicated. So how do you deal with that problem? So the solution to this problem is an enzyme that's called telomerase. Telomerase is not needed for the replication fork itself, so it's not part of the regular DNA replication. It's only at the end of the chromosomes. Telos in Greek is the end, the final goal. Remember telophase, right? That's the end goal, the last phase. Or the telomerase is the enzyme that finishes the end of the chromosomes. How does it do that? So let's take a look here. This is the top strand, right? We have the lagging strand. That's the problem that we have to deal with. The leading strand is fine. So here's the piece that hasn't been replicated yet because the last primer was just laid down here before that last piece was unzipped. So now what happens is that this enzyme, telomerase, brings a primer and it adds, so that this primer, these ends have sequences that are universal in organisms. So all the chromosomes have the same or very similar telomere ends. These are called telomere ends, the ends of the chromosomes. And so this RNA template binds to the end, to this end, and then the strand is extended out, not with anything coding for proteins, right? It's just a little bit of extra so that you can get a handle onto this strand that is now extended with these purple base pairs. The telomerase just adds on a little piece, and that little piece is good enough for a new RNA primer to be placed down and then be extended from the outside into to the inside of the chromosome, to the last end where that last primer was put down. And then after the polymerase runs into that last primer, we have the ligase, and that will then seal up the two strands. And that way we have dealt with that problem of otherwise having a shorter and shorter and shorter chromosome. So I write down here in a sentence, I'm not sure if that will help you or if the book has a better way for you to understand what's happening. I say telomerase uses an RNA template that fits the end of the chromosomes, which all have the same sequence. It extends the chromosome out further and allows the DNA polymerase to use that extension to synthesize the last stretch of the original chromosome. So that extension that gets added on is then used as a template to basically, from here on to here, right? That part is what we're after. This part is not coding for anything of interest, but this part is the part that we're after, that we don't want to lose. All right, so that is that's the story of the telomerase. I looked in your book to see if there was another animation for the whole process, because two-dimensionally it's kind of hard to understand, and I couldn't really find, I couldn't find one at all, but I looked online for something better or something at all, and I found a few, and I think this one is pretty nice. So I'm going to play this for you. Of course, you could also just watch it at your own time on YouTube, but why not watch it right now, and then we can go through the summary together. So
A is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so that's the animation. You may have noticed as you were watching it that in the animation, the lagging strand was on the bottom and the leading strand was on the top. That doesn't make any difference, right? Because you could basically just flip the DNA around and look at it the other way. So the point is not which one is the top and which one's the bottom strand. The point is that one of them, the leading strand, moves inwards towards the replication fork, and the other one, the lagging strand, moves outwards because it cannot grow continuously. All right. So let's take a look at the summary for today. We talked about the fact that DNA is synthesized in a semi-conservative manner, and you just saw that again in that animation at the end. The growing DNA molecule, like a growing RNA molecule, grows from its 5' end to its 3' end because that's the only way polymerases can add new nucleotides. Due to the anti-parallel structure of DNA, the two strands are synthesized in opposite directions. Right? That's because they run 5', 3', 3', 5'. That's what anti-parallel means. The two strands differ. One is the leading, one's the lagging strand, because one can grow continuously and the other one is made in sections. And those sections are called the Okazaki fragments. I list here all the primers, basically your toolbox that you need for doing this. RNA primers, RNA primase. Remember that this is an enzyme. The topoisomerase, helicase, polymerase, ligase, and the exonuclease. You also need nucleotides, just as building blocks, to start that process and actually fill in the new parts of the DNA. We talked about the fact that DNA polymerase has a proofreading capability, and that DNA synthesis starts at specific sequences called origins of replications, or ORIs. And then at the end, we talked about the problem with finishing the end of a chromosome, a linear chromosome as it's being synthesized. and how telomerase adds this little piece of extra template at the end, which can then be used by DNA polymerase to finish off that last piece of DNA. So that's what you need to know about how DNA is synthesized in the cell. Next time, we're going to talk about how this process with very similar enzymes is used in the lab to synthetically make DNA. And that's a very important process for all kinds of DNA technology. So next time we'll talk about DNA technology, and you will see that it essentially uses this process that is happening in the cell, just tweaked a little bit and made available for technology in vitro, meaning in the test tube. So we'll talk about that next time. Until then, I'll see you in class.